Thanks very much. I'm not an HIV researcher, so don't ask me any questions about exactly how these models can be used to study HIV, but you can talk to Hans Peter. We work on very similar types of models, asking somewhat different questions. So my interest is really as a hematologist and a stem cell biologist trying to understand hematopoiesis in a model that's relevant to humans, because there's many, many aspects of murine hematopoiesis that don't actually predict the behavior of human hematopoietic stem and progenitor cells, something I learned over my early career trying to do HSPC gene therapy and transplantation based on murine models that were not predictive. And also in a mouse, you can do limit transplantation to see how individual stem cells behave at a clonal level. But that's asking what that cell can do under incredible replicative stress. And as a transplanter, usually we don't want to do limit dilution transplants in humans or in monkeys, given how expensive they are. So we want to ask what HSPCs can do under more physiologic conditions, under the kind of conditions of autologous and allogeneic transplantation when you're giving thousands and thousands of long-term repopulating cells, and also to be able to study human and primate HSPCs. And I was also very interested in looking at issues of ontogeny and the behavior of individual cells over their lifespan, because there's a lot of interesting data coming out of single-cell RNA-seq and other approaches that suggest some of our classical models of hematopoiesis with bifurcation down very stereotypic intermediate precursors and progenitors may not actually be reflecting hematopoiesis, and maybe in some ways artifacts of the ways that we started sorting cells when monoclonal antibodies first became available. So we focused on using genetic barcoding to label individual HSPCs with integrating replication defective vectors. Originally this was done with murine Maloney leukemia viruses that have been gutted, but now all of our work is obviously done with lentiviral vectors, so HIV-based or SIV-based vectors that have been gutted and contain genes of interest or are clonal tags. So we can actually retrieve insertion sites as clonal tags, and that's been done extensively in human gene therapy trials, but that is pretty inefficient and not very quantitative. So about 6 or 7 years ago, collaborating with Rong Lu and Irv Weissman, we began using very high diversity genetic barcodes, random sequence barcodes that we put into our marking vectors that also contain GFP. They have a library ID, so we can use, in some cases, multiple libraries, multiple cell sources, or different things we've done, even within the same animal, as well as a very highly diverse barcode library. They can be retrieved efficiently and quantitatively after low-cycle PCR and Illumina sequencing. So this is just the model we use where we basically put our barcoded lentiviral library at basically relatively low transduction efficiency, because we only want to get a single barcode per cell. So we're targeting somewhere below 30% transduction of our engrafting cells, just a single exposure, and then we do autologous transplantation. Most of our work so far has been with total body irradiation, but more recently we're looking at alternative conditioning regimens such as busulfan or antibody-mediated immunotoxins, as Hans Peter mentioned, where we've also been working with Magenta and 47 Inc. And then basically you're just retrieving your barcode from very sorted cell populations, doing your sequencing. There's a lot of informatics that goes into this, and Dr. Keel already talked a lot about barcode kind of informatics, so I won't go into all of that, but you need to be very sure you have a highly diverse library that is sufficient to be sure, relatively sure, that when you have a barcode in a cell and you find it, that it has to have come from the same original transduced precursor cell. If you don't have that, you really can't use this model. So you can't be working with hundreds. You need to be working with millions of individual barcodes, or at least hundreds of thousands. And again, you want to have a low transduction efficiency, so most of your target cells have only a single barcode. It's not as complicated to deal with multiple barcodes per cell as it is to deal with multiple cells per barcode, but it's still important. And you end up with data where the quantitative barcode read fraction does very closely represent the fractional contribution of the clone. And a lot of that validation is given in some of the papers I list on the right there. And a lot of our code for doing all the informatics is available on GitHub, and I'm happy to send links to anyone. So the kind of things you're looking at is just looking for, say, at the abundance of individual barcodes or clones contributing to, say, granulocytes and monocytes, and basically, we find that you always have very similar read fractions or quantitative contributions in these two lineages, which from many other approaches are known to be very closely related in terms of precursor cells. And we can also look at bias histograms to ask, are there, if you just count individual clones, not caring how big they are, do they always contribute more or less equally, or do you have highly biased populations? The other thing we found very useful is just doing heat maps that basically group clones by their average contribution bioinformatically, and they're basically similarity of contributions among all these samples that we look at over time. And this is just looking at T cells, B cells, monocytes, and granulocytes. And this is focusing on the largest clones. You can do this any way you want, but we do a lot of setting a threshold to be sure that the clones we're looking at, given the sampling we're doing and the transduction efficiency, that we can be sure if a clone is there or not there, or reasonably sure that it's meaningful. And basically each row is an individual clone or barcode, and each column is a different time over since transplantation, and we can find evidence for rapidly engrafting and then disappearing committed progenitors that are different in each lineage that disappear by 2 months post-transplant, and they're replaced by multi-lineage clones shown across the top that contribute at relatively similar levels to all the different lineages. We also find late after transplant that you have T cell clones arising, shown in yellow at the bottom, that are not tracking in terms of size with the other cellular populations. And this generally represents, peripheral homeotypic expansion of T cells that occur, as we know, and can be tracked in other ways, such as with TCR. And so this is a very useful way of looking at various questions. And so one of the things we noticed, again, I wasn't focused on the innate immune system, particularly when I started these studies, but we found a very surprising finding when we started looking at mature NK cells in these macaques, CD56 negative or DIM, CD16 positive. And unlike the other lineages and even the putative immature NK cells that are CD56 bright, CD16 negative, where we have multi-lineage, very multiple thousands of small clones contributing across all these lineages, we found these oligoclonal, very enlarged peripheral clones of NK cells that did not correlate particularly with their contributions to other lineages. And this is just one example, and we see these T cell clones, as I mentioned previously, that we presume are due to peripheral expansions. And we can also look at pairwise comparisons against all the clones. Is this just the large clones? And we find basically the blue means highly correlated across all clones between lineages and time points. The green means it's not. And you see this very unique pattern for the NK cells where they correlate with each other over time, but not with the other lineages. We can look at clonal diversity by things like Shannon Diversity Index, and again, we see the same loss of diversity over time and very oligoclonal diversity and skewing found in this mature NK lineage. And we can look at how much of the NK repertoire that's marked or barcoded is made up by these highly biased, highly expanded clones, and it's really quite remarkable over time, with individual NK clones making up, up to 10 or 20% of the barcoded NK population. So, we wondered what happens over time with these clones, and with all of our other hematopoiesis, such as short-lived granulocytes, and in the immature CD56 bright NK cells, we see a population in red of the early clones that disappear, and then a stable long-term population where each clone is really, even the largest clones are quite small, stable over time. But with these mature NK cells, we see these clusters waxing and waning, going up over time, as if perhaps they're responding to something in the environment, similar to what we see later with T cells. And again, these animals got total body irradiation, so their T cell reconstitution is very slow. And we're now looking at what happens with busulfan, where we get much more robust and earlier T cell reconstitution. So again, it suggests a response to environmental stimuli. I don't have time to talk about it today, but we got some CMV negative macaques from Mitty Cower and the Tulane Primate Center, and used that model to show that we still have these expanded clones in the absence of CMV, but when we experimentally infect with CMV in the NK cells, we get new expanded populations as if they are responding specifically to an environmental cue. And we do show that rhesus CMV does reactivate following transplantation in CMV positive animals. We showed stable clonal segregation of these individual barcoded clones by specific KIR, which is really interesting given that people have called KIR panels arising after transplantation in humans with CMV reactivation as being pseudoclonal. And we show in this case they actually are clonal. There are not a lot of KIR reagents you can use in monkeys, but we've been collaborating with Bernard L'Enfant using MHC tetramers as a way to do this, and we found segregation of these individual clones stable for many months with specific here. And this has all been published in Science Immunology earlier this year if you want to look at it. I'm just giving this basically as an example. So we come up with a model for peripheral self-renewal of mature natural killer cells independent of ongoing production from hematopoietic stem or progenitor cells. We did a bunch of depletion studies in the paper to suggest that these are probably arising very early from short-term hematopoietic stem or progenitor cells, but that they can then persist and be peripherally expanding and proliferating. This also has been backed up by some work in some acquired somatic mutations that occur in humans that also suggest this population of NK cells is very long-lived. And perhaps this is a way of looking at the aspects of NK memory or adaptive NK cells that have been very interesting over the past 10 years. And we think these are probably epigenetically maintained, and we're working on trying to figure out how now. We have got a vector now where we can do single-cell RNA-seq and retrieve the barcode and an RNA-seq library from individual cells. Went back into a monkey last week, so hopefully I'll be able to figure out what is it about these clones that allows them to persist. Another thing we can look at is behavior of individual clones at a stem cell or a progenitor cell level. We actually transplanted aged versus young macaques with barcoded HSPCs. And there's been all this interest in this acquired clonal hematopoiesis or clonal expansion that occurs in humans, discovered in 2014, that is very common with aging and is associated with progression to myeloid malignancies as well as cardiovascular disease. And we see in transplanted aged macaques that we see these natural clonal expansions over time that are contributing at levels up to 10 or 15%, versus in young macaques, we basically never see these clonal expansions. So that's very interesting. We found that at least one of these clones here has a natural DNMT3A mutation, which is one of the mutations that's been most associated with clonal hematopoiesis in humans. We can also study geographic aspects of hematopoiesis, again in macaques where we can do lots of biopsying and comparing tissues, different tissue sites or tissues over time. So we can show direct demonstration of prolonged hematopoietic stem or progenitor cell clonal segregation in the marrow, which was quite surprising. Some cases up to a year or more. If we looked at the CD34 positive cells in the left bone marrow versus the right iliac crest bone marrow, we find they are clonally completely different. And even though these cells are proliferating incredibly quickly when they first go in, it really suggests that they're spreading in a localized manner and are not going out to the blood and back to another site. And then, not surprisingly, we can show local production of myeloid and B cells, the same clones on the right, and the same clones as the CD34 positive cells, and on the left, the same clones as CD34 positive cells. Anyway, so this is just another potential application of this kind of approach. Anyway, so this is just kind of a summary of the kind of things you could think about doing with this approach, which may or may not apply to some of the questions that you're asking in HIV. So we can look at, is the stem and progenitor cell population contributing in, equal ways to different lineages, lineage A and lineage B, or do you see, myeloid bias versus, say, lymphoid bias skewing, which in our model we did see with aging, which was interesting. You can look at clonal expansions, as I've talked about, so looking over time. And if you do see a clonal expansion with an individual clone, predominating in differentiated mature cells, well, is that clone also predominating at a stem cell and progenitor cell level, or exactly what level is that clonal expansion happening? Or are you seeing peripheral homeotypic expansion, as we think happens with T cells, and now we have, information that suggests it may also be happening with NK cells. And then finally, you can look at this question, which I think is a lot of interest to perhaps HIV investigators, and I'm collaborating with Jason and hopefully VEFA on some of these issues of what's happening locally in tissues, potentially with some innate immune cells, in terms of whether the population matches the population in the blood, suggesting that you have flux in and out and no local proliferation, versus finding clones that are expanded only in a particular tissue area, which suggests they've got to be proliferating there. So I think there's a lot of interesting questions to ask. So just in terms of the people that did all this work, Joy Wu, who is a postdoc and now a staff scientist in my lab, has led all the barcoding studies. Along with a number of other postdocs in the lab, especially Rocky Yu, who worked on the aging studies, and a whole group of bioinformatics, computer scientists, math major postbacs who came to the lab and were able to set up some of these pipelines for me. And then Bob Donahue and Sogun Hong has now taken over as the director of our non-human primate transplant and genetic manipulation facility. So thanks very much. Sorry, I didn't want to run out of time, but I guess I talked even faster than usual. So that way we have time for questions, right?
Yeah, there's time for questions.
No, okay, nobody.
Did you clarify everything?
Or I totally blew everyone away and they have no idea. Well, actually, all the barcoding work that was already introduced is helpful. And actually, Hans-Peter did some very nice work directly comparing insertion site retrieval to, the barcode approach, and it's definitely a lot more quantitative and a lot more robust. And hopefully now we can do it with single cells, which you could do with nanopore, et cetera, with insertion sites, but would be very challenging. Hi, Jonah.
Hi, Jonah Sash from OHSU. Lovely work. I love the idea of barcoding the host, and I wonder if you thought about combining that with the barcoding of the virus from Brandon, so you could barcode everything, and then you could actually really then drill down for reservoir studies at both the virus and the host level.
Yeah, I mean, that would be great if, especially once we can get to the point of using conditioning other than TBI, because I think, people like reviewers, again, all of you in the room are very sympathetic, except for the mouse guys. When they ask you, well, how come you only did 3 monkeys, and can't you do why haven't you done a secondary transplant to prove it's in a real stem cell? You can't do an ablative, 1,000 rad transplant on a monkey twice, for God's sake. Anyway, so there's a lot of, but these reagents of using these immunotoxins for conditioning, without getting, tissue damage to the kidneys and the lungs and everything else, and the thymus, most importantly, hopefully will allow, some of those kind of studies, and that would be super interesting. But right now, nobody's done that, to my knowledge.
About the peripheral maintenance of the NK clones, so you mentioned that they are maintained for months. What's
years.
So the longest that you have identified the same clones?
We have animals where we've followed for 5 years, and they wax and wane, and some of them are starting to disappear, the big ones early, but they have lasted up to 5 years in some cases.
So did you have the opportunity to see or recognize any precursors in the bone marrow of the
We cannot find any population in either the bone marrow, the lymph nodes, the salivary glands, the liver, all the places that people think that, NK cells mature in sort of these classical models. I think they probably do mature, but in terms of where they actually clonally expand, the only place we can find these expanded clones in terms of, not ruling out tissue, vascular that's just running through organs, the only place we find them is in the blood. So, it's interesting. We're still looking at that, but when we've looked in the liver, we've looked at many, and lymph nodes, we've looked at these tissues, and we don't see, overrepresented individual barcodes, in a node that we don't also see in the blood. So anyway.
So what is the model then, that these cells exist in tissues and recirculate periodically in the blood?
Yeah. And if you look in lymph nodes, the vast majority of lymph node NK cells are actually CD56 bright, 16 negative, and they definitely match all the other hematopoietic lineages and are turning over quite quickly. But we also did some, proliferation staining, et cetera, in the blood, and especially when you recover from depletion with a CD16 depleting antibody, et cetera, you get very marked proliferation of the mature NK cells in the blood. They can definitely do that, and we find the same large clones come back when we use CD16 depletion, which is very short term. When we use, in the paper we showed when we use the IL-15, very long-term IL-15 depleting antibody that Dr. Pickers talked about as well, that we wipe everything out. And when the NKs recover, you get new clonal expansions, but they're not the same clones. But we, from the KIR studies as well, we have individual clones that are persisting for years.
So even for, let's say, CMV infection, et cetera, you don't have overrepresentation of certain?
The CMV animals, we did not carry out tissue biopsies. The problem is getting those CMV negative macaques, as you can imagine, was A, incredibly expensive, and B, incredibly hard. And so we did not do any tissue biopsies that we thought could put them at risk of not surviving. Like, you know, so we looked at nodes, we looked in marrow, and we did not, you know, we didn't find any particular clonal expansions going on there. We've only so far found it in the blood.
Thank you.
These are all autologous because we didn't want to get into, I mean, Leslie Keene and Joda, I mean, I'd love to, you know, collaborate with somebody that is doing allos, but in our facility we had made, at least for right now, we've got plenty to do with gene editing and gene therapies and other things that we focused on that we've decided to not go there with allo. We're doing some CAR T work, but we're, you know, anyway. It would be interesting, obviously, to do that.